Thanks for joining me today. I'm going to tell you how to make a dichotomous chart. So if you look over here at the dichotomous chart page, we're going to basically learn what it is and then how to make one. Now first let's talk about this word, dichotomous. As it suggests, there is a split between two things. Dichotomous chart, okay? So basically, in the simplest form, dichotomous chart is something that is like a decision tree, okay? So you have some choice to make among 7 or 100 things. And what you do in order to make that decision is you ask a question which has a yes or a no answer. And depending on the answer, that narrows down the possibilities. So at each point in the decision tree, you ask a question that has a yes/no answer, and you narrow down the field of possibilities even further. So we're going to do that today. Now, first I'm going to show you a simple sort of generic example of a decision tree so that you understand what I'm talking about. So if we go to look at this page right here, I am going to have to make a decision. So my decision is I wake up in the morning and I can't decide what to have for breakfast. So I list the things that are available to me and I try to decide. So here I'm going to list them. I could have bacon. I could have eggs. Maybe there's orange juice in my fridge. Or there's some toast. Okay? So those are all my options. Now, the first thing I want to do is ask a question that's going to help me to narrow down these choices. So the first question I'm going to ask myself is, do I want protein this morning? Yes or no? So here's my first question. Protein? Yes? No. So if the answer is yes, then that narrows my options down to 2 things, right? Bacon, eggs. If the answer is yes to this question, now I have those 2 options. But if the answer is no, then we have orange juice and toast. Okay, am I done? No, I'm not done. I still haven't made a decision. I asked this first question, I have an answer to it, but let's, for each of these 2 possibilities, ask another question. So let's suppose I decided I did not want protein. Orange juice or toast, how do I decide between those things? Well, I could ask myself, do I want something hot? Yes or no? If I want something hot, then it makes sense to have the toast. Or, if I don't want something hot, I can have orange juice. Okay? Now, that then helps me to make the decision if I wake up in the morning and I want protein for breakfast, or I don't want protein for breakfast, but what if I do want protein for breakfast? So how do we figure that one out? We have our bacon and our eggs. How are we going to decide between them? Do I want meat? Okay, that would be one question. Now, I recognize that some of you are going to argue with me that eggs are meat too. So let's just say, for the sake of this argument, that bacon is meat and eggs are not meat. Okay, vegetarians might have something to say in there. Meat, yes or no? Yes. Then the answer would be, I'm going to have bacon for breakfast. No? Eggs. So this is a very simple example, but there is what your decision tree would look like. So let's apply this to a more relevant example in this course. Suppose that instead of what to decide for breakfast, I have to decide, or I have to devise a way to tell what microbe I have from a number of options. So, and this is, in fact, this is the assignment that you're about to do, is identify an unknown microbe. So let's look at an example here. So suppose that you have 5 possible microbes that you might receive. Okay, and what are they? Well, in real life they're going to be different than these ones, but let's just list 5 hypothetical microbes. let's say we have Bacillus nubi, Micrococcus lucy, S. morona, and E. coli, that's a real one, Staphylococcus aureus, also a real one. Okay, so here are our 5 options. Now, in the example I'm about to give you, I'm going to start with a question that is in fact not the first question that you'll use in your ID exactly, not when you're building your chart. So I'm going to come back to that, but it's just a nice way to show this as an example. So one of the techniques that you've learned how to do, and one way that you know that microbes are, or bacteria in particular, can be classified into one category or another, is by doing a gram stain. So if we decide to do that as our first step, we know there are 2 options. If we do a gram stain on our unknown, we might get a positive or we might get a negative result. So now we have to list which of the options that we have would be either positive or negative. So let's say M. lucy is positive, and S. aureus is positive, and S. morona also positive, and on the other hand, E. coli and B. nubi are both negative. Okay? So are we done? No, we're not. So we have to go to each of these different possibilities and we have to ask a question that's going to help us narrow them down. Okay, so let's start on the left. This is going to be a little easier. We only have 2 options. So what we need to do is we need to ask a question to which we get an answer that will tell us which one we have. Now, you know how to do this already. You have done a variety of biochemical tests and a variety of selective media and differential media to ask different questions about microbes. So you know how to ask if they can do fermentation of sucrose. You know how to ask if they can degrade a milk protein. You know how to determine whether or not they have the enzyme amylase or the enzyme oxidase. So in this example, we're going to put them on a milk plate and see if they can digest casein. So we're putting them on a milk plate, and the question we're asking is, can they digest casein? And we know the answer is either that they are going to be positive or negative on this milk plate. Yes, they can digest casein. No, they can't. So in this example, let's just say that this is how it breaks down. E. coli is positive on the casein plate and B. nubi is negative. All right, so now we have this situation resolved. If we have these 5 microbes and we do a gram stain and it's negative and we do a milk plate, then we have an answer. But we still have this possibility. We might do a gram stain and it might be positive. We still have 3 guys to figure out. So let's go to this list and figure out what test we could do that would allow us to distinguish between them. In my example, I'm going to do a nitrate test. What I'm asking is, can they reduce nitrate? And for the sake of argument, let's suppose that S. morona and M. lucy cannot reduce nitrate, but S. aureus can. Okay, so if we got a gram positive and it was nitrate positive, we would have an ID. But if it was nitrate negative, we wouldn't. So we still have to figure something out for these 2 organisms. If it ends up that one of these was actually the one you received, you would still have to have enough of a chart that would allow you to get to the bottom of it. Okay, so let's do one more test. And we could do another milk plate here if we wanted to. Just because there's one over here doesn't mean we can't do it over here. Okay, these 2 tests are under different contexts. This is a test we're proposing to do if it's gram negative. This is a test we could legitimately do even if it's gram positive. They're just different, okay? So in this case, we might get a negative, we might get a positive, and in our example, let's suppose that I'm going to argue that M. lucy is positive for casein and S. morona is negative. That, in a nutshell, is your dichotomous chart. So is this exactly what you're going to be doing? It's pretty close, but you're probably asking yourself, where do I get the information that I need? What kind of organisms am I going to be working with? Now, in the example here, these are 5 organisms that I wrote down, but those are not the ones that you could have. There actually are more than that. And so where you're going to find the information that you need in order to make your charts is online. It's 2 Excel spreadsheets, and there's one Excel spreadsheet for the gram-positive options and one for the gram-negatives. Okay, so I'm going to show you now on this computer where to go. So here we have your Canvas homepage. If you go into modules and into the appropriate week, the module for the week that you're doing this lab in this particular case, I believe we are doing it in week 8 or week 7. There it is. Okay, so in week 7, under Lab 5 information, you can see that there's something called the biochemical test keys, gram-positive and gram-negative. So let's look at that. Okay, so what you can see is that there's an Excel spreadsheet, and at the top of this page it says gram-negative rods. So there's an Excel spreadsheet with information about gram-negative rods. What is this information? Well, what you can see is that there's 1, 2, 3, 4, 5 gram-negatives over here, and on this column there's a list of all the different biochemical tests, many of which you have already done. In fact, all of them you should have already done. Okay, so we can see that there's a catalase test, indole broth, citrate, slant, nitrate, broth, starch, casein, glucose, sucrose, and lactose fermentation. And over here what you can see is for this organism, you can see the whole profile of its results. If you were to do a test on this organism, this is what you would see. So now it's a little bit more complicated than just this, and I'll get to that in a second, but what I just want to sort of say here is that if one way to do this, okay, would be to take every single possible test you could do and just do them all in your unknown and then compare that to the list of results here. Okay, you could do that, but we don't have the resources for that. I'm going to show you a way that's more efficient. So what we're going to do is make a dichotomous chart using this information. Okay, so what I'd like to emphasize at this point is that you need to make 2 dichotomous charts. Okay, you need to make one for the gram-positives, one for the gram-negatives. So we are going to be doing, as part of our unknown ID process, you're going to first, when you get your unknown, you're going to do a gram stain. That's the first thing you're going to do. And based on that gram stain, you're then going to either use your gram-positive dichotomous chart or your gram-negative dichotomous chart, which means you have to come to class with both of them prepared. So what is your chart for the gram-negatives? And you need a chart for the gram-positives. And as I said before, where you get the information you need is from the Excel spreadsheets online. Okay, so once you've done your gram stain, you're going to choose one of these charts, which means that you're not going to put the gram stain in your chart. We're going to start off with the assumption that we know what it is. Okay, and just to remind you, dichotomous chart, this is what it's going to look like, okay? You're going to ask a question. There'll be a yes or no answer. You're going to narrow down the options. You're going to ask another question, and you're going to keep going until you have identified all of the possibilities. So let's do this for example. I'm going to help you work through one possible version of a gram-positive chart. So gram-positive dichotomous chart, here we go. So the first thing we need to do is we need to write down the options. Okay, now you're looking at this screen and you're wondering, where are my gram-positives? Well, I'm so glad you asked me that. If we scroll down, and this is the one thing I just wanted to mention, see how this says gram-negative rods? Depending on the way that this posts in your Canvas, it might not fit all on one page, in which case you'll see this extra piece. That's also a gram-negative, so make sure you get them all if it's 4 pages long. So all the gram-negatives are up there. Here's our gram-positives, and again, there's a little bit of extra piece down here. So what are the gram-positives? I am going to do this just to make this simpler so that we don't kind of go crazy with the technology here. I am going to do this chart as if we're just dealing with these organisms. But when you go to make your chart, make sure you get them all, okay? So I'm just going to write down these 6 guys. M. luteus, Micrococcus luteus. M. roseus, Micrococcus roseus. Sarcina aurantiaca, S. aurantiaca. Staphylococcus aureus, Staphylococcus epidermidis, and E. faecalis. Those are all our possible options, okay? So we have to start by making a chart that's going to allow us to tell these guys apart. Now, I want you to look at this information here. And I want you to notice that some of the lines are grayed out. So let's look at indole broth. See how all of the organisms are negative for indole. So ask yourself this question. Would you do an indole broth as a test to try and narrow down which unknown you have? I hope the answer is no, because it wouldn't be informative. It wouldn't tell you anything. Same thing with the starch plate. Everybody's negative. Or if everyone was positive, that would also be unhelpful. Now, you could do any of these other tests that aren't grayed out. I'm going to recommend something. I'm going to recommend that you start with a test that you can get immediate results from. Now, remember, most of the tests that you're learning how to do, you have to inoculate and you have to incubate and you have to then come back, and sometimes you have to add reagents, so it's a longer process. But there are 2 tests that you learn how to do that only required you to do something. It really quickly gave you an answer. Do you remember what those were? Do you see them on this Excel spreadsheet? It's right at the top. So here's your catalase test. And what you'll notice is that 4 of these organisms are positive for catalase, 2 are negative. Okay, so let's do that as our first test. Catalase. We might get a positive, we might get a negative. If we have a positive, then we might have M. luteus. We might still have M. roseus. We may have S. aurantiaca. We might have S. aureus. Oh, and I misspoke earlier, so it looks like there's only 1 guy that's negative for this test, E. faecalis. Look at that. So if we were so lucky as to get a negative for catalase, we would have our answer right away. Are we done? Well, we're only done if we're so lucky that we got E. faecalis. We don't know what we get. Okay, so we have to make a chart that's going to give us options no matter what we have. So we have one way of identifying E. faecalis, but we've got these other 4 guys to figure out. And I am definitely missing one of them, so let me put him in. Okay, there we go. Now, if we have to distinguish between those organisms, between M. luteus, roseus, S. aurantiaca, and the staphylococci, what could we do? Well, there are a lot of options. Okay, we don't go back and redo the catalase test. That's not going to tell us anything. But we could, for instance, do, I don't know, maybe a nitrate. Could be negative, it might be positive. Okay, so if the nitrate is negative, then it's either M. luteus or S. aurantiaca. If it's nitrate positive, all the rest of the guys are in play. M. roseus, Staphylococcus aureus, and Staphylococcus epidermidis. Okay, so now let's come down to this part here. How do we tell the difference between M. luteus and S. aurantiaca? There's M. luteus, there's S. aurantiaca. We have to look at them and compare them and see what our options are. We have a lot, actually. We could do a urease. We could do glucose fermentation. We could do a citrate slant. But I like the idea of doing a urease test. Okay, and one of them is going to be positive, one's going to be negative. That's going to identify which which of these possible organisms you have. You still have 3, right? M. roseus, S. aureus, and S. epi to worry about. So you would still have to make a test for that. All right. So this is going to be, you have an ID, you have a different ID, but over here we still need another test with 2 options, plus or minus, and depending on how this rolls out, you're going to have 2 options that are positive and one's negative, or 2 that are negative and one's positive, and so for the one where you have 2 left, you still have to do something, right? Suppose we had 2 there and 1 there, we would still have to do another test. Now, hopefully that's clear. Are you done? Oh no, my friends, you're not done, because we still have to do our gram-negative charts. Okay. So for this, you're going to do the exact same thing, right? You're going to write out the list of the things you have. Okay. So I don't know, I'm not going to bother to write them all out, but I know E. coli is one of them, and there's a whole bunch of other ones. You're going to pick first test. And just like you did with the gram-positives, I had urged you to pick a test that gives you an immediate result. So look at that Excel spreadsheet. Let's go back up to the gram-negatives. And again, you'll see that you can't do a catalase as your first test. Why? Oh, think about why that might not be a good first test. But you could do an oxidase test. That gives you an immediate result. So let's suppose that's what we do. Either get a positive, we get a negative, we have some options over here, and then we go from there. Okay? And you just keep building your chart until you're all done. So depending on what profile of organisms you got for being oxidase positive, you would do another test, positive or negative, list the options. Over here, if you're oxidase negative, you would do another test. Positive, negative, list the options. So that's really, it's straightforward. Now, I want to say your chart may not look like your lab partner's. It doesn't have to look like mine, right? It's fine. Do it your way. If you don't like my idea of starting with things that give you immediate results, because you don't like the oxidase test when you did it, the colors were a little, I don't know. When you did catalase test, you didn't get bubbles. Whatever test it is you like, you can start with that. It's fine, okay? I'm suggesting the most efficient way is to start with a test that gives you a quick result, but it's up to you, and there's no wrong answers. The only thing is that you have to come to class with this already done, okay? So that is all the time we have today, and I appreciate your attention. I hope that you have fun doing this. It's really a fun exercise, and I assure you it will come in handy on the tests in the future. And I hope you'll tune in for another lecture series. In the meantime, have a fantastic day.